Assalamu alaikum. Sa'atum sabahan. Well, I would like to thank the organizers, Dr. Bazahouk and Hamid, for inviting me to speak to you here today. It's my pleasure to join you here. Last year, I was invited by the same team here, actually, almost a year ago, to speak about sort of gastrointestinal eosinophilic disorders. And I chose to end the presentation last year by sort of sarcastically saying to my colleague, well, I think you're going to go out of jobs. Eventually, all of you immunologists, sort of pulmonologists and so forth, you may actually come to a time where whatever we do in terms of different interventions, medications, or so forth, might actually get replaced by things like fecal microbiota transplantation. That's out of just, few cross-references, really. But I am pleased to say that Hamid actually took that into account and invited me this year to speak about, the relationship between food allergy and the gut microbiome, and how can we manipulate that, sort of studying the relationship and manipulate that in terms of treatment. And to start with, I might just actually, try and attract your attention to study this animal experience. And perhaps this is the most pivotal message I'm going to give out of my presentation today, is actually the conclusions of this experiment. And it might be worth just sort of looking or thinking carefully about those rats in the middle. These are actually germ-free rats. They kept in a special environment that allowed no bugs into their guts. And they were fed exactly the same sort of vegetables, so grass material. And they were sort of the environment they were living in were exactly the same. And what they did, then they brought in a group of infants, some of them with food allergy, established food allergy, others healthy children. And the food allergy was basically that of the cow's milk allergy. So the group of infants on the left side are healthy infants, and those on the right healthy, and those on the left are cow's milk allergic. They collected fecal material from them, mixed together, homogenized, and transplanted to those mice. These are actually germ-free mice. And then they did various studies on them, clinical observations, as well as laboratory, as well as histological examination of their alveolar specimens. So they studied the microbiota structure of their fecal material, the metabolites, whatever comes out into their stools, and then they examined exactly what happens to them after being sensitized to cow's milk protein in beta lactoglobulin, and then challenge them, to put them under a challenge with beta lactoglobulin. And what happened, surprisingly, those who were transplanted fecal material from healthy individual, they remained healthy. They were protected against cow's milk allergy. In contrast to those who were transplanted fecal material from allergic infants, they became allergic. They developed significant reactions which indicated the allergy status. And on top of that, they put a lot of efforts into different experiments. They concluded that actually out of the various species of those fecal material from the infants, they found this Clostridia species, the Amer cocci, as a specifically protective. So when I mean really protective, this is an important thing. It is actually protective against the development of cow's milk protein. So these clever people there, what they did, they divided the group of rats into three groups. One of them remained germ-free. So those in black are the germ-free ones. They remain germ-free. They did not transplant any fecal material into them. These are the mice transplanted with healthy fecal material. And these are the mice transplanted with fecal material from allergic kids. The drop in core body temperature indicates anaphylactic reaction in these mice, right? You can clearly see that the healthy ones transplanted with fecal material did not react. While the germ-free reacted, the cow's milk reacted. So here we are. So the healthy feces protected the development of cow's milk allergy. Those who did not have any germs in their bowel, the germ-free, also reacted. They actually showed anaphylactic reactions. And they went on further, and they tested the specific antibodies, the Ig antibodies against the lactoglobulin. Significant difference between the cow's milk allergic versus the normal ones, as well as they tested the IgE as well as the IgG. I mean, in rats, IgG is still of importance, and also the mucosal protease as well. So you can see from here that some fecal material provides protection. It's not just predisposing them, it's actually providing protection. And here they infused the specific bug, the Clostridia, the anaerobe stibes cocci. They infused it into the allium of some of the rats, and in comparison with the cow's milk allergy, the infusion of that specific material was protective. It was protective. So I think this actually created a significant sort of conclusion as, yeah, microbiota has got something to do with how do we deal with tolerance to food. So what is actually the gut microbiota? What is it that that they are actually claiming that it can be protective? So it's now called the virtual organ. The microbiota, the lump of the number of microbes, the structure, the genome, it's called the virtual organ because the influence it can have on the individual. And an adult, 1 kilogram of the weight is actually made of those organisms. Just imagine the huge number of them, right? 150 times, what is it? 100 trillion cells, 10 times the number of the human cells, 150 times larger than the human genome. You can actually imagine the influence that microorganism can have on the individual. So just to make it easy to follow what we're talking about, these are some definitions. So the microbiome is in reference to the actual organisms as well as the genetic material. And again, there is always a talk about eubiosis, which is the balance between the different organisms. Dysbiosis is the disturbances. And in that, they study the genes as well as the metabolites produced by these organisms, and called metagenomics or metabolomics. So there are thousands of these species, but mainly 4 dominant phyla: the bacteroids, the firmicutes, actinobacteria, and protobacteria. Now these nowadays, testing and studying these things is not really culture-based anymore. It's actually based on ribosomal RNA techniques, sort of next generation sequencing. So the most important fact to understand about the development of the microbiota is actually it established early on in life. Whatever happens in the first, say for example, 1 to 2 years, that's it. So the golden 1,000 days is really important. What happens, during pregnancy in the first 2 years of life is gonna be detrimental to your health in the future. Detrimental to the likelihood of you developing the non-communicable different chronic diseases, really. And that is why, you know, what happens early on is really important, and you come out without much in your gut, or perhaps some people say even there is some kind of transfer even into uterine, and it depends on the environment you're living in. You're going to develop a cocktail of various organisms there. As time goes on, this is a sort of dynamic and keep on changing. There are various factors which actually can affect the composition of the microbiota. You know, the mode of delivery plays an important role. Whether you are breastfed or bottle fed, the kind of food you're going to go on later on, the environmental factors of the food you eat, the antibiotics, the indiscriminate prescription of the antibiotics, and the various things. And accordingly, as time goes on, the composition of this microbiome keep on changing. But usually from here, first year, second year of life, that's it. It's finished with the kind of structure, what's going to be the diversity, the composition is fixed from there onward. So if you want to do something, do it early on. Right, later on, you may not be able to do much really. Yeah, so these are basically the effects. You know, if you were to breastfeed, you know, you sort of bottle feed, you change the chances of acquiring the normal commensals from the skin and from the breast and so forth. The diet, the family size, the hygiene theory, how careful we are about it. So what does this microbiota do? It does do various things really. You know, sort of it consumes or sort of it helps producing something out of nothing. The undigestible part of the food, out of that they produce, you know, vitamin K. They produce short-chain fatty acids, really important. Short-chain fatty acids, 3 of them. Butyric acid, butyrate is one of the most important, and the one you have to keep in mind. You know, these bacteria produces short-chain fatty acids, and butyrate in particular is of particular importance in immunomodulation and in the integrity of the intestinal barrier. Immunomodulation and the integrity of the intestinal barrier. Other biochemical functions of the bile duct circulation, mechanical protection or prevention against colonization by pathogenic organisms. How do they do that? It's actually the prevention against colonization. Mechanical things, you know, what do? They actually these bacteria compete with the pathogens, compete for food, compete for space. They don't give them a chance, right? And they create an environment which is not favorable for the other bacteria to live in. It reduces the pH of the intestinal lumen by the production of those fatty acids, and they produce antimicrobial compounds, you know, directly killing those organisms. And perhaps of importance to us is the modulation here of the immune system through the activation of the dendritic cells and the sort of anti-inflammatory effect of the IL-10, which I'm sure you know better than me, really, on the upregulation of the inflammation or down regulation. So that is the important thing. Again, mechanically, you know, what they do, they produce thicker mucous membrane or thicker mucus layer, tightening the junction between the epithelial cells and production of certain proteins like the beta defendants. So they're quite useful in health, but quite useful in they might cause disease as well. So yeah, butyrate or butyric acid is really of importance, one of the short-chain fatty acids. What does it do? Again, in thickening the mucous membrane, tightening the junction, and helping with the help of vitamin A, you know, and the production of retinoic acid and the combined effect on the dendritic cells. Eventually, you get excessive production or increased production of the IL-10, and that sort of modulate the immune function, and eventually acting on the plasma cells with excessive production of IgA and IgG, in contrast to the production of IgE on the other side. So butyric acid is really important. So if you lose the eubiosis, the normal balance between these bacteria, you go into a state of dysbiosis. So the balance is disturbed. One bacteria overgrow the other. And this results in dysregulation of the immune system and the various kind of inflammatory changes and the various different kind of diseases. Now, the microbiome is really implicated for all different issues at the moment.
5 minutes.
You know, the allergies, the obesity, and so forth. So we're going to just focus on the allergies here quickly. I'm sure all you know there's a significant rise of allergies over the last few decades, combined with the excessive use of antibiotics and sort of extra load of these conditions in the on the hospital. And this is attributed to the hygiene theory. You know all about the hygiene theory, initially thought to be because of list of the infections, you know, parasitic and equivalent, and then you see the difference between people living in poor hygienic conditions that are less susceptible to inflammatory or allergic conditions in comparison to those who are living in a better sanitary conditions. And this is attributed to the, again, you know, with the those infections, the anti-inflammatory effect of the IL-10. So when they come to sort of study this on humans, on patients, they found a sort of a difference in the gut microbiota composition, right? The actual composition, the actual metabolites or the different products. They looked at the classical, you know, they look at twins, monozygotic, dizygotic, you know, sort of living in the same environment, allergic against allergic, and they find a difference, clear difference between the composition and both different groups, allergic versus non-allergic, and difference in the metabolites. And various other studies, right, looked at the composition. So yeah, there is a difference. Definitely, there is a difference in the structure and the metabolites of those microbiomes. Well, are we able to come to a definite conclusion as to what particular change is there? Unfortunately, not. The studies are very heterogeneous. They looked at different things. So basically, yeah, there is no specific bacteria consistently associated with development of food allergy. Nevertheless, the evidence is there, right? And we know that dysbiosis precedes the development of the food allergy. Before you become allergic, if we were to examine the microbiota, we found that it is abnormal in comparison to normal individuals. And the critical time is early infancy. What happens in early infancy really determines what actually complications you might develop. So evidence is there as to the pathogenesis of microbes in the development of food allergy. How does that happen? Yeah, in simple terms, you know, if you were to live in the natural environment, you've got perhaps, you know, you may or you may not get the genetic predisposition, and you live in the natural, you know, sort of less clean way, exposed to animals, big family size, eating together, eating from each other, drinking from the same cup and things like this, natural breastfeeding, use of good food, you will be protected. In contrast to the other ways, you know, extremely careful about cleanliness, a lot of antibiotics, cesarean section and so forth, you get predisposed to allergy. And in simple way, as we said, the evidence points more towards butyric acid and its relationship to the downregulation of the inflammatory cascade. You know, the T helper cells, you know, suppressing Th1 and Th2, and the production of immunoglobulin A in particular. And if we were to look in more details, as from the right studies here, you can see, you know, basically the butyric acid and similar ones, as I said early on, with the use of retinoic acid, these are the mediators in the middle. So that eventually IL-10, in contrast to the allergic one, where it is IL-4 and the production of IgE. So that's in short the sort of pathology. What can we do about it? Yes, you may really need to look at trying and prevent it from development by normal delivery, breastfeeding, good food and abundant hygiene theory, or you try and treat it. So this is preventing it. Start from pregnancy, mother diet, early food. So then the microbiota and the mother itself get normalized. Baby comes out, inhales or starts decolonization with normal bacteria. With normal a healthy microbiome. Later on, well, use of prebiotic, yes, plenty of trials. Are we able to reach to a conclusion? Very weak evidence, really. So different societies, organizations, they're not able to recommend the use of probiotic as a mean of treating food allergy, unfortunately. Except there might be some evidence in preterms on those who were delivered by cesarean section. Prebiotics, forget about it, really. No evidence. There were some trials, but no evidence. Symbiotic, the mix of both prebiotic and pre and pro. Again, there is nothing to report. Fecal microbiota transplantation, yes, absolutely. In theory, that's something which can be done. But how can we do it? The selection of the material, the kind of the donor you can deal with, can it be done? Yes, indeed, it can be done. We tried it in one of our patients in the past. Different patient with inflammatory bowel disease. We collected the specimen. We go through rigorous sort of screening process. We process it in the lab. We deliver it sort of endoscopically and through a nasogastric tube as well. We can give whatever dose we can, and it's proven to be safe. This was tried in immunocompromised patient. Apart from sort of bit of high temperature, bit of raised inflammatory markers for 1 or 2 days, it was otherwise safe. It can be done technically, simple, really, not that difficult. What does the future hold? I think it's going to be personalized approach, right? There will come a time where, at early infancy, so you can study the individual fingerprints or the genetic prints of the microbiome of each individual, and you can find out whether it is eubiotic or dysbiotic, whether it is within the normal of what expected or not, and then accordingly you sort of decide whether to interfere and treat or whether to just try and maintain. Are we there? Not really. We're not there. I think there is a long road to go, really. So in summary, yes, there is a definite relationship between dysbiosis and food allergy. Yes, we have actually looked at some evidence. Yes, it precedes the development of allergy. The most important part is first 6 months of life. Yeah, certain, it can be specific, species specific, certain items, bacteroides, clostridia, aneurostibes, cocci, and things like this might be the reason. No exact evidence as yet to the prebiotics. Future, yeah, it's gonna improve things, but perhaps this takes us back to the famous say by Hippocrates that all disease come from the gut, and if you look after your gut, you're gonna be healthy. This sort of attracted further attention, and yes, let them eat dirt. They will be much healthier. Thank you very much.
Thank you very much, Amer. That's very interesting indeed.